As scientists continue to study disease states in a variety of tissues, an understanding of the 3D structure of organs is providing insight into structure-function relationships that affect tissue functioning. Advanced imaging techniques are giving researchers a better view of the inner workings of complex tissues, but some significant obstacles still stand in the way of truly comprehensive tissue imaging. One such obstacle, the inherent opacity of tissues, has proven particularly difficult to overcome. Ultimately, if light can't penetrate a tissue, there's no way to image it. Scientists have searched for a method to turn tissue more translucent while maintaining its structural integrity. With the invention of tissue clearing, and notably the clarity technique, it is now possible to achieve true volume imaging of intact tissue. By virtue of the purpose they serve, lipid bilayers create barriers that make it difficult to image deep within intact tissue, specifically keeping the tissue opaque and milky and causing light scattering. Lipid bilayers also create diffusion barriers that prevent small molecules from moving freely within tissue samples, which in turn possesses a challenge for techniques such as immunostaining. But lipid bilayers are such an integral part of tissues that removing them while keeping the tissue intact for imaging has not been possible until the creation of clarity, which works by selectively removing lipid bilayers from tissue. Clarity transforms intact tissue into a hydrogel tissue hybrid construct that is perfectly intact and mechanically stable, as well as permeable to small molecules and photons. Hydrogel cross-links secure biomolecules, making it possible to remove lipids from the tissue without disrupting its structure. Clarity starts with a hydrogel monomer infusion into the tissue of interest. The tissue sample is incubated in a hydrogel solution to enable complete diffusion of the hydrogel into the tissue. After the tissue sample is fully infused with hydrogel, it's polymerized, which then cross-links biomolecules into their location to completely preserve the molecular structure of the tissue. After polymerization is complete, lipids are removed from the tissue to clarify it. This is performed either passively or actively by placing samples in a detergent solution. Passive removal of lipids is less costly, but it's not a reliable method, especially for large tissue samples such as entire organs. Active removal of lipids from tissue samples is faster and more efficient. Active lipid removal occurs via electrophoresis. Samples are placed in buffer containing SDS and the current is applied across the sample while the buffer is continuously circulated. The exact time required for electrophoretic removal of lipids depends on a variety of factors, including the current applied and the temperature of the solution, but it can range from several days to several weeks. After clearing, tissues lose their milky white appearance and are ready for volumetric imaging. In addition to being optically transparent, cleared tissues are permeable to macromolecules, and small molecules and biomolecules, including antibodies and RNA, can permeate the entire tissue. Although clarity gives researchers unprecedented access to tissue imaging, there are some common difficulties scientists may face with electrophoretic tissue clearing. Poorly constructed clearing chambers with inadequate buffer circulation can leave bubbles trapped in chambers, causing poor temperature control and inadequate clearing. When the tissue sample is overheated due to a number of variables such as poor temperature control, irregular electrical currents, or inadequate buffer circulation, damage to the tissue can result in burning, melting, and the formation of black precipitates. The conditions that cause these problems can be very difficult to correct manually. To address these challenges, commercial systems for performing clarity are available. These systems provide an all-in-one solution that takes the guesswork and protocol development of the process and enables researchers to clear tissue for imaging rapidly and consistently. The use of commercial systems also reduces the amount of time clarity requires, from up to 2 weeks for a mouse brain to now just 8 hours. Studying the structural and functional relationships of complex tissues remains an important field of research. With the application of clarity, scientists are able to gain unprecedented insight into tissues and disease states.